we have seen the formation of the male gamete and the female gamete. The last we saw was the formation of the female gamete or the egg cell. Now further, it is pollination which has to occur for fertilization to take place. Now what is pollination? Pollination is the process wherein the pollen grain is deposited to the stigma of the flower. Now this pollination is of two types. First is self-pollination and second is cross-pollination. Let us look at what is self-pollination. Self-pollination is when the pollen grain is deposited onto the stigma of the same flower or a genetically similar flower. So the flower is either same or it is genetically similar. Further, self-pollination is again of three types. The first one is called as autogamy. Autogamy means when the pollens are deposited to the stigma of the same flower. For self-pollination to occur, the pollen release and the stigma receptivity have to be in synchronization, means they have to occur around the same time. Now, in autogamy, since it is taking place in the same flower, the pollen grain is deposited onto the stigma either by bending or by growth or by folding. So that is autogamy, means it is occurring in the same flower. Now second is geitonogamy. Geitonogamy is when the pollen grains are deposited to the stigma of another flower but which is genetically similar. Now this autogamy can be further divided into three types. First is homogamy, second is cleistogamy, and third is bud pollination. In homogamy, it is again that the pollen release and the stigma receptivity have to be in synchronization. It is occurring in the same flower. And here also the methods that would be followed would be bending, by bending, growth, or by folding. In cleistogamy, this is occurring in closed flowers. Homogamy has occurred in open flowers. Cleistogamy occurs when the flower is closed. So the chances of self-pollination is very high. There is only self-pollination that can take place in a closed flower. So only self-pollination can occur over here. The pollen grain has to be deposited onto the stigma of the same flower. Bud pollination, the pollen release and the stigma receptivity, they occur before the bud opens. Before the flower opens, the anther has matured and so has the stigma. So once the bud opens, they are both ready to exchange. So the pollen grain gets deposited to the stigma of the same flower. So this was about self-pollination. Let us look at cross-pollination. Now, cross-pollination is when the pollen is deposited to the stigma of a genetically different flower. So the plant will be different. So it's deposited on a genetically different plant. Now, since the exchange of the pollen grain to the stigma of the flower is on a genetically different plant, so it needs some external agents to carry the pollen grain. Now, these external agents can be either water or wind or insects, animals. Based on this, there are types of cross-pollination. Now, the pollination which is carried out with the help of water is called as hydrophily. Now let us look at hydrophily. The water is the carrying agent which carries the pollen to the stigma of another plant. Here, how will the character of flowers be? The flowers are not attractive because it's water, so flowers need not be colorful. They do not have a strong odor. The nectar produced is very less here again. But the pollen grains, how will they be? The pollen grains are light, they are small, so that they can be easily carried by the medium of water. How will the stigma be of these flowers? The stigma is sticky so that the pollen grains stick to the stigma. So that was about hydrophily. Second, let us look at pollination by insects, that is called as entomophily. Now here, 80% of pollination by insects is carried out by bees. Now for the plant to be pollinated by the insect, it has to be very attractively colored. So the flowers here are very attractive to look at. The fragrance is strong. Lot of nectar is produced. The pollen grains here will again be light in color. The pollen grains here again will be light in weight. So the majority of the pollination by insects is carried out by bees. Third, let's go to pollination carried out by the wind. That is called as anemophily. These flowers will be called as anemophilous flowers. Now, because it is pollination occurring by the wind, the pollen grains produced will be very large in number. Since they are very large in number, there will be high wastage. So, one of the drawbacks here is that the wastage is high. And it is through wind pollination that we see the pollen allergy that takes place. Here, the flowers are again colorless or maybe very faintly colored. There is no strong fragrance to them. There is no nectar which is being produced. The pollen grains, how will they be? Now they have to be carried by the wind. So they are very light, they are small, produced in large numbers. How will the stigma be? The stigma will be either dry or it is mostly sticky. The pollen grains are a little hairy so that they can get attached to the stigma. They are hairy, light, produced in plenty, and the stigma is sticky. So that was about anemophily, that is wind pollination. Then we come to pollination carried out by the birds, that is called as chiropterophily. Now chiropterophily is when pollination is occurring by bats, when bats are responsible for pollination. Another type is ornithophily. In ornithophily, the agents responsible for pollination are birds. So when birds are the agents for pollination, the flowers are again colorful and attractive, lot of nectar being produced. One important type of pollination is zoophily. Zoophily is when the agent of pollination are animals. Here, the flowers are again scented, colorful, attractive looking. And the last type of pollination is malacophily. In malacophily, it is the snail which is the agent of pollination. So now we have seen what are the 7 types of pollination, of cross-pollination mainly, and also we have seen the types of self-pollination. Now at the end of pollination, we can realize that self-pollination is a source of less variation. Means the more the self-pollination that occurs, there will be less variation seen in the flowers and the plants, and because of less variations, the level of evolution will again be low. So cross-pollination is a source of producing more variations and better qualities. So I can say that self-pollination, if continues, it produces an inbreeding depression. So for that reason, we should see what are the characters in flowers which will promote cross-pollination. So let's see what characters in flowers promote cross-pollination. First is dichogamy. Now, dichogamy means when the pollen release and the stigma receptivity are not synchronized, means they are not occurring at the same time. Now, because they are not occurring at the same time, it is cross-pollination which will occur. Self-pollination will not be able to take place. So that is called as dichogamy. Second is the placement of the anther and the stigma. So the placement of the anther and stigma is such that the pollen grains cannot be deposited onto the stigma of the same flower. Third is something that is called as self-incompatibility. Now this self-incompatibility is occurring at the level of genes. So this is actually a genetic mechanism, or I could say there is some genetic mechanism which prevents pollination by the same flower. It prevents fertilization to occur by the pollen grains of the same flower because it's a genetic mechanism. Next, I could say unisexual flowers. So if the plant has unisexual flowers, there are more chances of cross-pollination. There could be no autogamy. Detenogamy could occur. Cross-pollination can also occur, but there is no autogamy which is taking place. Next is the length of the style and stigma. If there are different lengths of style and stigma, there is again more probability of cross-pollination occurring. So that is called as heterostyly. So heterostyly again increases the susceptibility of cross-pollination. In bisexual flowers, if there is a physical barrier between the anther and stigma, then too the self-pollination will not be able to occur. It will cross-pollination which will occur. Also, similarly, in the bisexual flower, if the anther or the stigma are suppressed, that means if either of them is not allowed to express, that means either the male gamete or the male gametophyte or the female gametophyte are suppressed, it will lead to cross-pollination. The self-pollination will not be allowed to occur in the same flower. So that was about how to decrease the chances of inbreeding depression. So these are called as outbreeding devices. How can we promote, or how do the flowers promote outbreeding? We have seen how pollination takes place and the types of pollination. Before that, we saw the development of male gamete and the female gamete. Now that the pollen grain has deposited onto the stigma of the flower, the next step that takes place is fertilization. So here the male gamete from the pollen grain will fuse with the female gamete which is present in the embryo sac, which is present in the ovule, present in the ovary. So here the fertilization that takes place is called as double fertilization. Now let us see why is it called the double fertilization. Once the pollen grain reaches the stigma, the pollen tube enters the style. It passes through the style because it is releasing enzymes which will make passage for it. At the same time, it is absorbing food from the style. The pollen tube reaches the micropylar end and it enters the ovule. So if the pollen tube is entering through the micropylar end, it is called as orogamy. If the pollen tube enters through the chalazal end, it is called as chalazogamy. And if the pollen tube is entering through the integument, through the sides of the integument or any other point, it is called as mesogamy. Now this type of fertilization which involves passage of the pollen tube through the style is called as syngamy. Once the pollen tube enters the ovule through the micropylar end, it comes across the synergids. We had seen that the egg apparatus has one egg cell and two synergids. So now that the pollen tube has entered, it will come across the first synergid and that synergid gets destroyed. Now after the synergid gets destroyed, the pollen tube will release the contents. So what does the pollen tube have? It has two male gametes. So it releases the two male gametes which are haploid. Out of this, the first one will fuse with the egg cell or the female gamete. So that's the haploid male gamete fusing with the haploid egg cell to form diploid zygote. So this is the first fertilization. This is also called as generative fertilization. Now we have the second male gamete which travels further, and if we remember, there is a central nucleus, there is a secondary nucleus in the center of the embryo sac which was diploid. Now with this diploid secondary nucleus fuses the second male gamete which is again haploid. So now the product that is formed will be a triploid. Now this triploid nucleus is called as primary endosperm nucleus or the PEN, P-E-N. Now this is the second fertilization that has taken place. That is why this entire process is called as double fertilization. Now this primary endosperm nucleus will further on development form what is called as the endosperm. It is the endosperm which will further provide nutrients for the zygote to grow and so on. So this is the entire process of double fertilization. The second fertilization here is also called as vegetative fertilization. So this is how the process of double fertilization takes place. Now let us see what are the types of endosperm. There are basically three types of endosperm: nuclear, cellular, and halobial. In the nuclear endosperm, it is only nuclear division which is going on taking place continuously. There are many nuclei which will be formed from the nucleus of the endosperm. The nuclei then get pushed to the periphery because a central vacuole is being formed. Then the cytoplasm begins to thicken so that the vacuole becomes smaller in size. But at the end, there will be only nuclei in the entire endosperm. There are no cell walls which are being formed. So there are no separate cells. It's just full of many nuclei. Second, cellular. Here, the nuclear division has taken place. After that, cell walls will be formed so that there are multiple cells in the endosperm. Third, in halobial. Now, halobial is an intermediate between the nuclear and the cellular. Here, after the nuclear division has taken place, the cytokinesis will also take place and so on. The first cytokinesis, that is, the first cell will be formed, and after that the nuclei will be free. So that is about the halobial endosperm. In halobial endosperm, two chambers will be formed. The micropylar chamber will be the larger one.